scientist and the colleague who is always very generous to give a time to Pakistan despite his busy schedule in his research group and the various groups he is interacting and collaborating all over the world. And his personality actually does not need much introduction, and most of us know. And he's a Professor Monir Iqbal from the Fulbright Institute. He has a long-standing career on protein biochemistry, and in addition to this, he has developed his career, I would say, the expertise in the vaccinology, and particularly on the poultry vaccines, influenza virus vaccine, and DB vaccine. And he is, I would say, is a long-standing scientist at the Fulbright Institute, and his director, Don King, is here as well. No, you are a director. No, not yet. You are a group leader. Okay. So anyway, with this introduction, I will request Professor Monir Iqbal to enlighten us about improving the efficacy of poultry vaccine, which is the problem in every type of the vaccine, and it is very important to address the efficacy of the vaccines. Thank you very much, sir.
Thank you, Lord. Many thanks for inviting me and give a chance to share some of my work. So, as was said, I am Monir Iqbal, working at Fulbright Institute. So, my group, the main focus is, I would say, three things. One, how the poultry viruses, particularly avian influenza and Newcastle disease, evolve to persist in poultry, and what are the changes these are the determinant they acquire to increase the virulence or the persistence in poultry, and the third thing, how we can control it to improve particularly vaccines and the diagnostics. So here I am only talking about the vaccines and the new technology we are trying to use to improve the efficacy and potency of these vaccines. So for example here, there are several poultry viruses which are causing disease to the poultry. For example, avian influenza, Marek's disease, and so on. There is several chicken anemia, pox viruses. And loads of, for example, this is a schedule is largely being used in Pakistan or many other countries. Loads of vaccines to these single bird is being given on different days. And if you see that more than 40 doses and in some cases 50 doses of vaccine are given in a 1.5 year during the life of a breeder flock. And even first 3 weeks you give 3 or 4 of Newcastle, and still there is a problem. It's not like you are giving the vaccines and the problem has been gone. The problem still remains, but is it just reduce the losses. So we need improve these vaccines. One way to give, rather than 40, we give much less, maybe 4 or 5 rather than 40, to protect the bird from these viruses. So in both cases, either the vaccine has a more potency, so you only give 1 dose, or you have a multivalent. So the challenge is why we are currently giving a large amount of vaccines or the large number of doses. The reason is that one of the reason is the virus is evolved and the vaccine efficacy goes down, and the virus continue, either we are using the vaccine. And the new viruses are emerging, and these are the virus variants, and if the vaccine do not match to your field viruses, then of course your efficacy of vaccine goes down. And then the co-circulation of multiple variant, again the same. If you have the vaccine against one variant, the other variant within the same, for example, in Pakistan like H9 NDU, IBV, so other continue, and you say that you have given the vaccine, but still the virus is continue. It's not the vaccine problem, is the problem is the variant continue to circulate. And then, of course, there are some viruses which are called immunosuppression, and that's your vaccine efficacy goes down because the bird, the host is not responding to your vaccine because it has immunosuppression. The other major issue is because of the antibody you have already in the bird through maternal-derived antibody or the previous vaccine in older birds. And then, of course, when you give the vaccine, some of your antigen is being blocked by the antibody, and therefore it do not work. And then it comes to the poor quality, and poor quality means that you are giving a vaccine, but that vaccine do not match again to your the antigen. Yesterday, Don exactly said that your quality of the vaccine is there. If there is no quality, then probably you are giving just a water rather than the vaccine. And that is, and sometime I heard that the people use half dose. If the dose is a recommended one, so you have to dose a full dose rather than the half dose. So all these practices has to compromise the efficacy of your vaccines. So what is desirable? If I give a vaccine, I want the vaccine should be potent, and a single dose should induce faster, stronger, and durable immunity. And if possible, if the multiple antigen is there against the multiple pathogen, again, it's effective. So it's not only protect against the clinical disease, but also reduce shedding and transmission. So the less in the environment, the virus load in the environment reduce down. So therefore, of course, the vaccine is there. So if less virus, you have more protection. Against it is for the poultry. This point is affordable. So we can make the vaccine which nobody can actually use it, so that's not the issue. So the people can actually afford it and can use the vaccine, so it has to be cheap and it's easy to deliver, particularly in poultry. And then of course they should be safe. We can use a live H9N2 or flu vaccine, but we are not allowed because that's not safe because it's due to ressortment and so on. And also stability. If we make the vaccine, we have to be stable at least. We can keep it at least 1 year or more. And in UK, another country, the DIVA strategy should be adopted for avian influenza, so we can detect or differentiate between the infected and the vaccinated animals. For going further, what you want out of it if you give the vaccine ultimately, and that is a BOHA requirement for the flu vaccine, which I don't know when it will be possible, but that is ultimately we can get a freedom from the disease, and there is no trade embargo if you are doing vaccination, and then also there is no effect on the production of, so if you give the vaccine and ultimately the production goes low, so then there is no use of that vaccine. So there is no effect onto the production or any other serious effect onto the birds, and ultimately reduce the indemnity, so the disease burden from the environment goes less and less. So currently the vaccine which are for the poultry are available, or mainly these two, and now subunit are coming. So the inactivated virus vaccines, mainly we can say that field viruses you can select, but if you have hundreds of viruses, you need to select which one is the best. or in our case, we can generate the recombinant viruses by reverse genetic, but the minimum for the production is at least they can replicate in eggs or cells, depending on which virus you are using, at least in term of flu, at least 10 to the 9 or NDV should be replicating, and then you can actually use for the production. Similarly, for the vector vaccine, again, we are using herpes virus of Turkey and Newcastle disease and duck enteritis virus. Used to be work on foulpox and IITV. So these are the only vector which are being used or potentially being used for the viral vector vaccine for the poultry. And then subunit vaccines, there are one or two commercially available, one I know only, but there is a potential that these subunit can be available. The only thing is that it should be cheaper than producing any alternative way, for example in eggs, and if it is cheaper and it is then certainly the subunit has a much more benefits than using eggs or anything else, or even the whole inactivated virus. There is a special talk, but I do not have a time for that. And the DNA RNA vaccine, again, now the technology is coming, and hopefully if we can afford these vaccines for 1 penny per dose, then of course that can be also in the market very soon. But the only thing is for the poultry it has to be extremely cheap. But each vaccine has its own benefits as well as challenges. So benefit I already told. The challenges is, for example, if you have making the whole inactivated virus vaccines, the field viruses may contain some hidden pathogen. So you have the field virus and you may have say that the majority is let's say a flu or H9, but it may have some other viruses which you need to purify. So you do not want that something else is in your vaccine seed. And then the field virus also should replicate in high titers, and it has a more kind of a broadly or at least strong antigenicity. So that is and therefore you have to use repeated doses for the conventional vaccines. Attenuated, again, it may revert to the higher virulence in some cases, and that is a problem because you have just attenuated by passaging in the cells, and if it comes back, passage into the animal, it may revert it. Again, viral vector vaccines, again, there are a couple, but the major problem with these one is the maternal drive antibody. If the virus do not replicate in the host when you vaccinate, what happens is that because of the maternal drive antibody, it will not produce the required amount of your antigen, recombinant antigen, therefore less immunity or delayed immunity. Subunit vaccine, again, they are effective, but in some cases, if you have a single antigen, it may not be protective. Luckily, flu and Newcastle can, but there are some viruses which you need a multiple antigen as a subunit rather than a single antigen. So that production of multiple antigen for a single disease sometimes it is a problem. And then again, mRNA or DNA vaccine that certainly require more innovation to make it cost effective. So therefore the new innovation is required, and we are using reverse genetic or CRISPR technology to engineer the virus, which can increase the safety of the vaccines, which I cover later. Safety means, for example, high path virus we cannot use for production. We need a high containment, and for high containment, the expense to maintain the high containment facility is more than actually producing the vaccine. So again, your vaccine should induce a much stronger and broader immunity. So for that one, you can select in vitro or nowadays on computer which antigen you want. You can have a consensus sequence, and then you can use this technology to make the antigen and make the vaccine. Again, similar way we can actually put antigen into the viral vector by consensus sequence or changing or so many ways, some of I will cover, and that we can express even multiple antigen in a single vector. And then for subunit, we can engineer cells or the vector to express more. So this is the one which I am producing, not the antigen, but the antibody, so we can provide a passive immunity. So one first thing which I said that reverse genetics, so we use for in this case avian influenza virus as a reverse genetic. So these are the background which draws more out of genes, 6 genes which provide the virus to replicate, and these 2 genes are for the antigenicity. So any virus, if you have a sequence, so you only put these from the field virus, and then you can generate the virus in the lab. And when you generate this virus in the lab, so it is, first thing it is a clean vaccine, so it do not have any field contamination. And secondly, these genes can, in this way we can modify, for example, removing the polybasic cleavage site, which I cover it, and you can increase the safety, and then you can actually modify it to increase the antigenicity to match the field viruses. Again, increase the breadth of immunity and increase the vaccine replication, which I now cover each one. For example, here, this is a high pathogenicity avian influenza. This is a marker, this is a polybasic site in the HA. What we do is, instead of these one, when we reduce to the low path virus, which has only a single arginine, what happens is this virus, which is a highly virulent to the chicken, become less virulent, and not only to the chicken, to the embryo. So when we make the vaccine into the cell culture, so we can harvest a large amount of virus, whereas if we do this virus, it kills the embryo immediately, so the production level or the yield of the virus will be less. And also this virus, high path, is also zoonotic, therefore there is a risk to the human as well to the environment. So the whole waste you generate by, let's say, 1 ton or thousands of eggs, you have to dispose of more, spend more money and a facility to actually dispose of compared to dispose of which is low path. So the risk assessment is less for this compared to the low path. Similarly, you can identify that which of the amino acids are more critical to compromise the vaccine efficacy. For example, in this case, we identify, and this is published, that 180 and 183 amino acid in the H9HA are very critical for the immunogenicity of these vaccines. So for example, if the wild type virus in the field has A amino acids, the vaccine will induce a cross-reactivity against the homologous 1024. When the virus has at that position T or V, then it's compromised the vaccine efficacy drastically. Similarly, at that position, if the field virus has N, it is a same kind of quite good immunity. But if the field virus contains now D because of evolution or mutation, then it compromised. So you need a vaccine which, even because in the field the virus contain this, the variant contain T or V, and then the wild type has N, and the variant co-circulating at D. So in Pakistan currently All these mutations are in viruses. Some has A, some has T, some has V, some has N, some has D. So you need a vaccine which cover this as well as this, and this as well as this. So that is sometimes difficult to isolate the virus from the field, and that has that opportunity. So you have to select a virus or generate a virus which can cover all these mutations. And this is example, one of the example that this is the virus, the vaccine we make having A at this position 180, and it's a very high immunity against homologous. We can see HI titer of 12, whereas the viruses are circulating in Pakistan which have either T or in this case T and N. We saw that these viruses are not cross-reacting if we have a vaccine seed which contain A. So we need a vaccine which has which cover these two. So we selected a vaccine antigen which has T at that position, and we raised the anti-sera, and then we saw that all three viruses, different variant, this vaccine is highly cross-reactive, at least giving a 10 against all of them. And that means that if we use a seed like this, and this is very easy by engineering a virus and introducing mutation, rather than looking into the field hundreds and thousands of viruses. Similarly, we then change make some changes in antigen. In this case, we make the antigen and put a glycosylation in the H9HA at that position, and that changed the electrostatic interaction with the receptor, and that increased the antigenicity. For example, we have this vaccine which is homologous cross-reactive like this, but when we have this vaccine which has T plus this mutation, we drastically increase the immunogenicity, and by increasing the immunogenicity you can also increase the broader, because if there is some variant come which is a less cross-reactive, still this can cover. So this means that by reverse genetic and the data which is published in the literature, you can use it and you can generate a safe virus which can be used as a vaccine, and that is easy by reverse genetic rather than looking for the field. The other problem for the vaccine yield is the amount of virus you select from the field, and now it is not replicating enough. So you want to reduce the eggs used, and therefore the virus you are using as a seed should replicate enough to make the vaccine doses as much. For example, here we have a virus, this UDL Pakistan virus, it replicates here. But if I use the egg, I can make from one egg 100 doses. But if I change it, because I found a virus, for example, in this case a Vietnam virus, which has a higher replication, and we identify which gene make this virus more replicable, and that was the PB2 gene, and we introduced that gene into this virus, blue virus, and then the same virus, because HA is the antigen, the same virus now in the egg we can get 1,500 doses. Because of 10 times more replication or 1.5 times more replication, so this is the increase is 1.5 log extra, and that 1.5 log 10 increase make a huge difference for the producer. Similarly, we want to check that what is the best vaccine which we can select for the field. So for that one, for example, H5N1 or H5NX viruses, last 10 years or 20 years, they are evolving and they are changing. So rather than you have hundreds of viruses and raise the anti-sera, you can do on the computer to find out the representative strain of each clade. In this case, this is H5 clades from 2.3.2 to currently 2.3.4.4H. So you can have representative of each, and this is only 20 or 22 viruses rather than you have, for example, thousands of viruses. So the data from thousands of viruses you can reduce to 20 viruses, and then you raise the anti-sera against this one, and then you do the whole analysis with that virus and the anti-sera, and you see that which one is the best for target in your field. If the target is, for example, 2.3.4H, this is in China mainly, so you can select that particular vaccine. For example, here, this vaccine, if it's a homologous, it gave 11.7 log HI titer, but the H5N1, which was then emerged, that do not cross-react, the cross-reactivity was only 0.9. If you use this vaccine against the field virus, which is this vaccine would not work. So therefore, you need to do the background, all these analysis, and it is very difficult if you do thousands of viruses. So you have to select, because a lot of sequence data is available, so you can use that sequence data and then generate reverse viruses, and then you can do this kind of analysis. And we normally do to more representative. For example, this is the virus H5N6 virus in China, and the same clade, and this is current virus, which is the clade currently H5N1, circulating in UK and Europe and mostly all over the world. And if we have this vaccine, it won't work against this, and if we have this vaccine against, it will not work against. So this is a visual one. We can see which strain we should select for a particular country or the region. We also using Newcastle disease as a vector, and also how to generate a Newcastle disease which can be antigenic match to your field viruses. So what we do is, for example, this is a lentogenic strain like Lesotho, and then what you see is the main antigen is F and HN. So from your local strain, you can see what is the homology of your local strain. So you can take F and HN and introduce in there, and then this virus is the same virus almost, which is circulating in your poultry. But the problem is this virus, which is, so currently we call it genotype 7, this is highly virulent, so you cannot use a field virus, so you have to remove, similar to the hypath virus, the polybasic sites from the F protein, so make it lentogenic, because if it's a mesogenic or the virulent velogenic, you cannot use, because it again killed the embryo. So it's the same story. So you modify your F gene to make the vaccine, and then that vaccine you can use against Newcastle disease. But if you want to make it a multivalent, you can introduce, in this case, H9HA or the double H9 and H5 and so on. So in this way, you can actually modify the virus by reverse genetic, and you can make the vaccines which are homologous to your local strains as well as the multivalent. So the antigen you can put also from the local strain. And this is one of the examples. So we have this virus. So we vaccinated the bird, and then we've done HI titer by giving a single dose to 7-day-old chicks. So we saw a reasonable antibody, a high antibody against the Newcastle disease genotype 7, which is circulating in Pakistan. Similarly, we inserted the H9, and we see the antigenicity against H9. And this way, a single vaccine, a single dose can protect against Newcastle disease and the H9 avian influenza. We also produce HVT, which we either put 3 antigen or at least 2, is quite, means the gene, the virus replicate efficiently as the wild type. So when we put NDVF or H9 or H5, so we can detect and we can vaccinate the bird and we can induce immunity against NDV or H5 or H9. And this is in cell culture when we infect the serum we use to detect the virus. And here in this case we do the H9. And this is NDV antigen and this is H9. So that means the virus is expressing the protein of NDV as well as H9. So this HVT can protect against AIV, H9, NDV, and Marek's disease. And this is one of the example when we given to the chicken. So this is H9 and anti-sera after three weeks. And then because F protein, we have to do ELISA. So that ELISA results are equally competent and quite good. We also modified IBDV, one of the work of the colleague. So the problem is the vaccines. Okay, so I move on. So we also produce this multivalent vaccine by subunit, and we also just modify the antigen by putting the antibody, and these antibody goes to the cells and induce rapid immunity and overcome maternal-derived antibody. Examples are here. So we given the vaccine to chicken which has a very high maternal-derived antibody, and you can see after 3 weeks, even 3 months, the antibody titer of the target one is more than 10. We also produce the antibody which we can do spray, directly passive immunization. And basically this is my, just the last thing is the conclusion. So basically you can implement innovative approaches which can enhance the efficacy and the duration of the immunity. And of course, your vaccine will be cost effective and reduce your losses. And this is my team. Thank you very much.
Can I have 1 or 2 questions, please?
So, Professor Morir, so what do you think? Which viral vector you will prefer, herpes viral vector or the NDV, as far as maternal antibodies of both viruses in the chicken?
Yeah,
these experiments have been done
currently, if you want to use a live vaccine, and in your field there is a maternal-derived antibody, then HVT works alive. But the problem is that the HVT replicates slowly. So it takes some time, 4 weeks or even 6 weeks, to induce optimal immunity. And for that one, the company advised that you also give a killed vaccine. So the question farmer asked, why should we use it if we have to give a killed one?
Yeah.
Okay. So, but the HVT do work. It overcome maternal because the mechanism is different. But the only thing is you have to improve that you do not have to do an extra dose.
So may I have a question?
Sure.
So as your presentation, you were mentioning the mutation recorded in case of low-pair H9 in Pakistan. But in our case, there are a lot, I think more than 20 types of vaccines are being imported from different countries in Pakistan. My question is that, do you think this use of multi types of vaccines from different countries are triggering the mutation chances in Pakistan, or what are the potential factors where it is?
I don't think that is the issue, that whether it's come from other country or made locally. If they are inducing immunity, yes. and that is protective, then any vaccine can work. And you only have to see whether that vaccine is inducing immunity against your local, whether it is any company. So company is not the issue, it's the induction of immunity.
Okay, so Dr. Manu, can I ask you a question?
Sure.
Yeah, we are curious. You have a lot of the research on the bird flu virus. Maybe it can be a challenge. Also challenge the work globally. And you know, a lot of the manufacturing, lot of research, they develop different approach against the bird flu virus. Can you give some good idea how to reduce the bird flu virulent strain in wild bird?
In wild bird. So
Yeah.
the question is how we can reduce the virus from the wild bird.
Yeah.
Yes. So the lady, we have a. I will take 1 minute more. So Anne is the. I think she is a princess. Princess Anne came to our institute to open our laboratory, and she asked the same question. That is okay. You can protect the poultry. What about the wild birds? Because she is interested to protect the nature, to protect the. I would say it is difficult, but you can reduce if you have a reduction of your virus in your poultry. Because the virus is going from poultry to wild bird, it's not only one-way traffic. So you would reduce it, and the nature will decide that one, because if, for example, H5 and 8 was there, and what happened is the virus push it, because of the natural immunity, the virus cannot survive longer. Same we are seeing for the current H5. In 2022, it was havoc, and since October 23 till now, we only have 5 or 6 outbreaks in UK, but the same period last year, we have hundreds of outbreaks. The reason is either the virus change or the host push the virus to change naturally in wild birds. So if we can reduce the virus prevalence in poultry, I am sure we can protect them. I am not saying that we can start vaccinating to the wild bird. What we are saying is if the environment is clean, we will reduce.
Okay.
Thank you so much. Please allow me to introduce our next speaker, Professor Sir Andrew Pollard from University of Oxford, UK. A distinguished leader in the field of healthcare and medicine. With a career spanning decades, Professor Pollard has left an indelible mark on public health, serving as the director of Oxford Vaccine Group and making significant contributions to vaccine research and development, particularly in the fight against COVID-19. Recognized for his outstanding achievements, he has been honored with the prestigious awards such as Knighthood for Services to Public Health and the Oxford University Vice Chancellor's Innovation Award. Beyond his professional endeavors, Professor Pollard is an avid mountaineer, showcasing his adventure spirit and determination. He is one of the most famous scientists in UK after COVID pandemic. He was supposed to join us in person, but he has to go for a WHO mission in Uganda. And from there was network issue, so he has generously sent his presentation in the form of a video. He will be giving his presentation in two parts. First, he will be speaking about typhoid vaccine currently being used by NIH, and also later on he will tell us about the preparation of COVID vaccine at war footing during the pandemic. So please give a big hand for Sir Andrew Pollard.
is most common in the Thank you so much, Mohammed, for asking me to speak to you today on typhoid vaccines. And I'll talk a little bit about some of the work that we've been doing here in Oxford. But actually, with you in Pakistan, some of the world-leading work on typhoid and typhoid vaccines has been going on over a number of years now. And it's therefore a great pleasure to be able to talk about some of our experiences with an audience who knows so much about this condition. Now, typhoid, as you well know, particularly affects young children and has the highest rate of infections among school-aged children, and is most common in the cities in South Asia where there is poor access to high-quality water and rather limited sanitation opportunities. And that really is the ideal situation for the spread of these bacteria. When we look from a global perspective, as you can see on this heat map, in South Asia is where the highest rates of typhoid and paratyphoid fever occur, with the darkest colours on my slide here. There is also typhoid across the African continent. Interestingly though, paratyphoid is not really an issue in Africa for reasons that we don't understand. Instead, non-typhoidal salmonella are rather more common in that setting. So in the region where you're sitting in today in South Asia, then typhoid and paratyphoid are really the major causes of enteric fever. Now, as well as that burden in children, there's also this particular cause for concern, which is the spread of antibiotic resistance in typhoid. And this just shows some of the spread of resistance from South Asia around the world during the course of the last decade or so. And these were strains which were resistant to most antibiotics. And you'll all be well aware of the outbreaks of extremely drug-resistant typhoid in Karachi and elsewhere in Pakistan over the last few years, with some spread of those cases to other parts of the world as well. And these are strains which are resistant to conventional antibiotics, but also resistant to cephalosporins, with really only carbapenems and azithromycin as antibiotics that could be used for treatment. So the first thing that we did was to investigate the burden of typhoid in 3 different countries, and those 3 countries you can see on this slide are Malawi, Nepal and Bangladesh. And in red across the top, we tried to estimate the incidence by undertaking surveillance for typhoid in our populations. In each of those countries, we visited 100,000 individuals and got them to agree to us to monitor them over the course of the next couple of years to identify cases of typhoid happening that presented to the healthcare system. And what we were able to show from that was that the typhoid rate was rather lower in Malawi than in Nepal and Bangladesh, so lower in Africa than in South Asia. And the highest rates of typhoid occurred in school-aged children, which you can see very clearly in all 3 countries. And that is a pattern that all those who work in the region would recognise, much rarer in infants, more common in school-aged children. And this gave some really very high results with, between 200 and 600 per 100,000 person years. So these are very high rates of infection in these populations. But we were concerned that some cases are missed by this approach of just looking at what's happening in the hospital. And so first of all, we adjusted these rates for healthcare seeking behaviour. So to explain what I mean by that is, in Nepal, if your child has a fever, you're actually more likely to go to one of the pharmacies around the corner to get some antibiotics, rather than to make the journey to the local hospital, where you'll have to pay more and likely will get the same treatment. In our population of 100,000 people in Nepal, there were 1,000 pharmacies in the community, so very easy access to a community pharmacy for antibiotics. So we can make adjustments to the incident based on behaviour. Secondly, we found that paediatricians in many of these settings were unlikely to take blood from very young children. So we had an underestimation of positive blood cultures in the youngest age group, so we had to make adjustments for that. And finally, we find that the rate of sensitivity for typhoid is only about 50 to 60% on blood cultures, so we also made an adjustment for that. And by doing this, we found extraordinarily high rates of typhoid, so often more than 1,000 per 100,000 person years. And these seemed unbelievable rates of infection with these adjustments. So we also did some serological work to try and identify whether we could really believe these results. And the serological work is to look for seroconversion before and after infections in the population by taking 2 blood samples in a random sample of children in the population. And what we found from this is the rates of infection on serological grounds, so evidence of infection on production of antibodies, suggests that these adjusted rates are probably in the right ballpark. So a much higher burden of this disease causing febrile illness in children than we had ever appreciated by just running hospital surveillance. So the first vaccines for typhoid were developed in the late 1800s by Ormroth Wright, who you can see here in this photo. And he developed a heat-killed typhoid vaccine, which was given to British Army recruits who were fighting in the Boer War in South Africa at the end of the 1800s. The problem with this vaccine was it was very reactogenic, and the soldiers didn't like to take it because they felt unwell for several days afterwards, and they started to refuse to be vaccinated. Fortunately, that vaccine was more widely used after being reformulated by another army doctor called David Harvey, and was used successfully during the First World War. Now, on the background of several typhoid vaccines being used for travellers here in high-income countries, there was a sense that maybe for these settings where there's very high transmission, vaccines could be part of the solution to the burden of disease. And about 10 years ago now, the World Health Organization suggested that a human challenge model might be useful to help accelerate development of these vaccines. So a human challenge model is where individuals are deliberately infected with typhoid to allow testing of vaccines. And the way this works is that first of all, we grow the bacteria in a laboratory, and then we ask volunteers to drink a solution of bicarbonate containing live bacteria to try and mimic the effect of exposure to contaminated water, as might happen in cities in Pakistan or in other parts of South Asia, or the consumption of contaminated food. And so these are volunteers who, about 50% of them are students here at Oxford, the other 50% are other, usually professionals, but members of the community, adults in the community. And by drinking the bacteria, we can then monitor the individuals over the course of the next couple of weeks. And what we find is that about 2/3 will then go on to develop classical typhoid as you would see it in your clinic in Pakistan. So This is the fever chart here, and you can see it in black. You can see the temperature rising above 38 degrees in this individual who had a drink of typhoid on day 0, and by day 9 has a fever. In green is the C-reactive protein, which has risen, showing that this volunteer has got evidence of typhoid infection, and some of them have had rose spots as well. If we look at the cultures, there are some bacteria in the stool shortly after ingestion, but by the time the fever goes up, this individual has positive blood cultures. You can grow the bacteria from their blood, and they also are shedding the typhoid bacteria in the stool. And so we then treat them with antibiotics, and they recover from the infection. So this shows that you can set up a model of typhoid, and we found that by giving about 10,000 bacteria, we were able to infect about 2/3 of the individuals who had a drink of bicarbonate with typhoid in it. So that established a model that would allow us then to test vaccines. And here you can see a typical study where we vaccinate individuals with a typhoid vaccine, either the one used for travellers or the new typhoid vaccine that now has been rolled out in Pakistan, and then monitor the individuals closely for the next month, taking blood samples, next month taking blood samples. And then after a month, when they've made an immune response to the vaccine, we ask them to drink the typhoid bacteria to see whether they're protected. They either develop acute disease or they are protected. We follow them up after that with lots of blood samples to monitor infection. And with this study, we were able to show that the new vaccine that had been developed prevented typhoid in more than 50% of cases, and built confidence in the use of this new vaccine for further testing and rollout in countries with a high typhoid burden. And the data from this human challenge study were absolutely pivotal in supporting the World Health Organization's pre-qualification of the vaccine in 2018, the policy recommendations from the World Health Organization, which is to vaccinate children between 6 months and 15 years of age in high burden areas, and finally for the Global Alliance for Vaccines and Immunization to release $80 million for funding to help countries introduce typhoid vaccines. So this was a hugely important study to get things rolling, and many countries started to apply for funding after this for typhoid vaccine rollout, and as you'll know, Pakistan was one of those. But we were left with the question of how best to implement the vaccine in settings with a high typhoid burden. And this consortium between Oxford and the University of Maryland was set up to test typhoid vaccines in about 100,000 children in a randomized trial in those 3 countries where we've been undertaking surveillance: in Nepal, Bangladesh and Malawi. And this study got underway in 2017, and most of the 100,000 children were vaccinated over the next 6 months or so, and included those from 9 months to 15 years of age. The studies were very successful. It showed that the vaccine prevented about 80% of infections in all 3 countries in South Asia and in Africa, which was incredibly good news. And the success of those trials has led to further rollout of the vaccine. So this is the 2-year efficacy from Nepal in Kathmandu, published in Lancet Global Health, and you saw those other publications there. Bangladesh, very similar impact, about 80% protection there also, and very encouraging data from this study, particularly in Bangladesh. There were enough individuals in the youngest age groups to show that even in 9 months to 2 years of age, the vaccine had a very similar protective effect to that in older children, around 80% across all ages. And so that really was a critical story, suggesting that rolling out these vaccines have a huge effect. And now around 7 countries have introduced the vaccine, and rollout continues with many others planning to introduce in the near future, including, as I mentioned, Bangladesh. Next year, Malawi has already introduced, Pakistan, Nepal, and Zimbabwe, Liberia have all introduced the vaccine. And I think one of the most astonishing stories was that of the rollout in Pakistan, which occurred in February of 2021, when most of the world was completely focused on the coronavirus. In Pakistan, you were able to vaccinate around 1 million children per day during the first half of February, which I think is one of the most extraordinary achievements in vaccination ever. But that's the typhoid story, with rollout happening in many countries, the huge success of rollout in Pakistan, I've already mentioned. There's clearly more children still to be vaccinated in many countries, including in Pakistan, with more mop-up to occur. But the impact we're seeing in countries that have introduced is already very clear. But it does leave this problem in South Asia of paratyphoid, which is about 1/3 of the cases of enteric fever. And the typhoid vaccine does not cover paratyphoid. So we've started work here in Oxford on a human challenge model to test new paratyphoid vaccines. And we've just completed the first of those studies with a live oral vaccine. We don't have the results yet, but they should be available in the next month or so. So that's the human challenge for a live vaccine. But there's also new vaccines being developed in India, which will provide protection against typhoid and paratyphoid as an injectable vaccine, very similar to the typhoid vaccine that's already been used. So the future may include bivalent vaccines, but they will have to be developed through the challenge model for paratyphoid, because it's considered by the World Health Organization that it's not feasible to run the field trials needed to develop a paratyphoid vaccine. So we're working hard to support vaccine development in the hope that we'll be able to show over the course of the next couple of years that we can also prevent paratyphoid infection. But I think it's clear to all of us that vaccines should only be used for typhoid and paratyphoid in the interim, whilst we're sorting out the provision of clean water for everyone. And I think that has to be more of a priority for authorities in countries, because it would not only solve the problem of typhoid and paratyphoid, but of course all of the other enteric pathogens that are spread by contaminated water. So that's all that I'm going to say today. It's been a pleasure having the opportunity to speak with you, and just to celebrate the huge efforts in Pakistan and other countries where vaccines have been rolled out against typhoid, protecting children today. And thanks to the team here in Oxford, the many trial volunteers, our funders, and our colleagues in Nepal, Bangladesh and Malawi, who's helped work with us on all of these programs. Thank you very much.